The broadcast is now starting. All attendees are in listen-only mode.
Good morning, good afternoon. Thank you for joining us for today's webinar. Our webinar is titled Dotmatics Vortex, Supporting Chemical and Biological Data Analysis and Visualization. Our presenters today are Dr. Rob Brown, VP of Product Marketing, and Dr. Tedman Ellers, Senior Application Scientist at Dotmatics. Our audience will be on mute for the duration of the webinar. If you have any questions, we invite you to put them into the question panel, and we will answer them at the end of today's presentation. The recording of today's webinar will be available for download on our website within a couple of days. So now I'm going to hand over to Rob and Tedman for today's presentation.
Okay, good morning, good afternoon, good evening, everybody. Thank you for joining us. Here's the outline today. So we're going to start with an introduction to Vortex, which is the product that we're talking about today. And then I'll walk through some examples of what our customers have spoken about to try and explain to you how it's used. We will then walk through 4 example use cases, 2 for medicinal chemists, 1 for high throughput screening, 1 for bioinformatics. At that point, Tedman will give you demos of how those work. So Vortex can be used standalone to load files and analyze them. It can also be used in conjunction with the Dotmatics Informatics suite. And so the next part then will give you a couple of examples of those. 1, loading corporate data from the database rather than files. That's using a product called Dotmatics Browser. And then the second one, to use a product called Reaction Workflows to enumerate a library and then send it to Vortex for interactive analysis. And then I'll come back and finish up with some of the future plans, the things we're working on for the next release. So as an introduction to Vortex, what is Vortex? Well, as Tina said in the introduction, it's a product to let you visualize and analyze scientific data. So at the core of the product is the visual display that you see in the top graphic there, with interactive tables and grids of data, and then connected charts and graphs to let you see different views on that data. Once the data is loaded, then you can refine the data set either by filtering on interesting property ranges or activity ranges, depending on the data you have, or you can make selections. And as you see in the graphic on the top right, we've selected one slice of the pie chart, and that's then highlighted the equivalent rows in all of the other charts and tables. One of the things that really distinguishes Vortex is the fact that it's natively scientifically intelligent. So it not only understands numeric data, but it also understands what a molecule is, as you see in the top 2 charts, but it also understands then what a sequence is, as you see in the bottom 2 charts. It not only understands those scientific objects, but it has the analysis to go with it. So there is cheminformatics analysis, such as the R group analysis you see in the middle left, and you'll see in one of the demos today, and the sequence alignment analysis that you see in the middle right, which you'll also see in one of the demos. So as you load your data, it's often necessary to add more data to let you answer the questions that you want. So the data set that's loaded can then be augmented with scientific calculations, molecular properties, for example. Vortex can be used as a standalone application. I can load my data, I can do my analysis, get my result, and move on. But it's also often used between teams and in collaborations. And then you start to use things like templates and shared workspaces to have standard views of the data and to allow one scientist to create a view and then share it with others. One of the things that's necessary for the kind of data sets that we're all dealing with is to have high performance on very large data sets. So we can, within Vortex on a standard laptop, load millions of rows by thousands of columns, potentially with a very large sequence object or molecule object in each cell, and then still have interactive scrolling and interaction with the data. Vortex itself is open. There's a lot of calculations and charts built in, but there are always other things that individual customers would like to add. So there is a scriptable API that power users and IT teams can use either to create your own custom analysis or to integrate other third-party code that you have. So for example, very common examples are integrating additional property calculators, such as those that you might get from KNIME or Pipeline Pilot or something like that. It's a cross-platform product. We're going to show you on Windows today, simply because we have Windows laptops, but it also works perfectly well for those of you who use Mac or even Linux workstations. So that's a very brief overview of what Vortex is. Another interesting question then is, well, what do customers typically do with it? And one way to explain that is to go back through our recent user group meetings that we have around the world and think about what the themes were for the talks that were given on Vortex. And for those of you who are existing customers, if you have a Dotmatics support login, there is an archive of all these presentations on the support site, so you can go through and find the presentations behind these. But really what I've tried to summarize is what are the common use cases. So as I said, the most common use case is I want to do an ad hoc data analysis on a personal data set that I'm using in my research. It's the most common use case. And in a lot of cases, what customers have talked about is it's an improved process over what they had before. So that very often is we used to analyze our data in Excel. Sometimes it's been we used Vortex to replace tools like Spotfire or Surat. Moving beyond the personal, there have been a number of talks at the UGMs about using Vortex as a project dashboard. The idea here is that you would use standard templates with standard views and populate that with real-time data from the project. So any team member at any time can open Vortex and see exactly the state of play of the project data with a common view. And those templates can be company-wide or they can be project-specific. So they're simple enough to build that you can build specific projects, specific templates that address the concerns of specific projects. An extension of that, which has been talked about, is also using this then with CROs. So these dashboards can be used internally equally with the right infrastructure. They can also be shared so that scientists in collaborations that cross companies can also see the same dashboards of data while making project decisions. We've had a number of talks talking about the use of Vortex on the antibody discovery side for doing bioinformatics analysis. We have a product in the suite called Dotmatics Bioregister. This is the registration repository for all kinds of biologics, including antibodies. And so a number of customers have talked about the use of Bioregister with Vortex. Some of the use cases that have been mentioned are using Vortex to QC data that's coming in from CROs before it gets uploaded and deposited into Bioregister for the sponsor company to use. We've had talks about formalizing a bioinformatics system from a company that was basically operating out of multiple very large Excel spreadsheets. And by bringing in Bioregister and Vortex, they've managed to organize and secure all that data. And then in terms of the analysis, we've also seen talks where that replacing Excel and paper and shared files with Vortex analysis has allowed them to go from interrogating the data taking days to taking minutes. The whole of Dotmatics is an open platform, so we've also had talks where there's been another biologics repository. In the case I'm alluding to here, it was gene data. And again, Vortex was used in part of this process to pull data out of gene data, and then during the selection and panning process to walk through the data set, picking the antibodies for follow-up. And then in a related case, we had a talk using Vortex to analyze mass spec data where they were looking at antibody drug conjugates and trying to understand the loadings of the payload onto the antibody, and using Vortex for that analysis. On the screening side, this is one of the examples you'll see in a few minutes. Vortex can be used to do very detailed analysis of high throughput screening data, and it's also been used in one case to exchange screening data with CROs. For those companies doing DNA encoded libraries where you need to do the deconvolution of the hits, Vortex we've had a couple of talks about using Vortex for that. And then finally, as you'll see when we get to the demos, the primary views in Vortex are 2D. It's not a 3D modeling environment as such, but it has been used to present back to chemists both 2D and 3D data integrated. And in one talk that we had, that integrated PyMol to visualize the 3D data. Now within Vortex, there are a number of native 3D viewers for molecules and proteins as well. So that just gives you a sense that it's a very flexible tool. There's a lot that can be done with it, from the simple personal analysis all the way through to some very complex team projects. So now we're going to go through the 4 use cases. I'll introduce each one. And again, these are not comprehensive. They're just designed to give you an understanding of the breadth of the product and what can be done with it. So I'll go through each one of these, and then Tedman's going to demo them. The first one is very simple for profiling compounds as a medicinal chemist. And this is partly to allow us to actually introduce the product to you and see the basic capability. So as a chemist, I'd like to understand the properties of a set of compounds that I have of interest. Some of the properties may be in my database or in the file in this case that we're going to load, but others may need to be calculated. And then having got all my data together, I'd like to select an interesting subset for further processing. So in what you're going to see in the demo now, Tedman's going to import a set of compounds with experimental data. Then he's going to calculate some additional molecular properties. And then he's going to apply filters and select a subset with an interesting profile that we would then take to whatever the next step of follow-up is. So over to you.
Thank you, Ron. Okay, so what I'm going to do here is just drag and drop a file with some SAR data. So you can drag, drop files as well. And what we see here is a data table with our chemical structures, associated data that was captured in that file here. If we expand this, we can see that there's hierarchical columns here. You can drag, you can drop, you can sort the columns based on a particular property as well. You'll also notice that you can fix columns or fix rows if you'd like to have those references. Now, each column corresponds to a filter, so you can filter the table here. So the first column here, structure, allows me to filter based on a particular chemical substructure that I sketch. So here, if I just say BR, excuse my resolution, and then now I can see my selected substructures within my data table here. You'll also notice that it realigns based on those substructures as well as highlights those. Now we've got a number of properties here. And if we'd like to add additional properties, we can do that as well. So let's go into our tools. Let's calculate some additional properties. Maybe we can add some hydrogen bond acceptors, donors, a Lipinski cutoff. Let's get the percentage of sp3 carbons, and maybe down here, Marco scaffold with attachments. So this is using Vortex calculation capabilities. As Rob mentioned, you can also integrate other calculators if you'd like. And if we scroll to the right, we can now see that we've added hydrogen bond acceptors, donors, as well as some additional data. Multiple structures can be existing on the same row, as well as structure fragments. So this is nice. We've also got the capability of quickly binning our data. So for instance, for target 1, our PIC50, I've got several bins here. So if the PIC50 is between 8 and 9, between 0 and 5, and this allows us to do things like create categories of activity, so that if we want our PIC50 for target 4 to be high, and we can add secondary sorting capabilities to say if I'd like one category to be sorted high to low and another category sorted low to high, for instance, if we're looking for selectivity. Okay. So in addition to manually sorting, we can also create profiles. So in this case, I'm going to load up a target activity profile. So this is going to give me an idea of I'm interested in my target 4 being a large value, my target 2 being a lower value, and maybe some additional information there as well. So if I calculate this, it's going to create an activity profile score that I could then use to sort, as well. Okay. Yeah. Additionally, if I'd like to add maybe an ADME profile, I can add the ADME profile as well. Okay. Now, if I'd like to see what my data set looks like visually, I can add a chart. Right. So here, let's do a similarity visualization. Here, I'm going to use a chemical fingerprint to determine which molecules are more similar and display them in three-dimensional space. So here's a 3D plot of our molecular data here presented in three-dimensional space. So if I select some compounds in the chart, I can view those and see that those are indeed similar, right? Orthofluoros here, N-methyl on those as well. If I'd like to add additional data to this chart, I can configure the chart, right? Maybe my color should be my PIC50 for my target of interest. I could do a log-based coloring there. If I want to see what the actual molecular data is, I can add a tooltip and create a custom tooltip with my structure, and maybe I want to add some of my CYP information onto that. So now as I select compounds within that chart, I get the various tooltips appearing there. You know, all of this is designed to make your life easier. So if you'd like to export something to an image or to a PowerPoint slide, you can do that directly. You could just export this as either a PowerPoint or an image and be on your way. Okay.
All right, so if we jump back to the slides. So that's a basic compound profiling, and it also just shows you the basic capabilities of loading data, charting, graphing, making selections. So let's move on to a slightly more sophisticated medchem example. So now again, as a chemist, I have a set of molecules that share a common core. I have measured activity data for those molecules, and I'd like to understand how varying the substituents at different positions has affected the measured activity. So the Vortex solution that you're going to see now, we load the structures with their activity data. We're going to use the R group decomposition algorithm to identify the substituents, and then we're going to create a 2D SAR plot to understand how the activity varies with the combination of substituents. So the end result will be what you see over on the right-hand side. This is an example where there were several R group positions, more than 2, but what we've plotted here is R1 versus R2 substructures. So we have a range of activities depending on what the R3s and R4s are, but what we're looking for are those with high activities irrespective of what the 3s and 4s are. So pie charts with more green in this case. Okay, so let's see that one live.
Okay, so let's use the same data set here. And what I'm going to do now is do an R group analysis. I've got a history here of R groups that I've used previously. If I'd like to modify this, I could. Let's just use R1 versus R2 for this example. And here I'm going to run that analysis. So there was 82 molecules that had positions there at R1 and R2, and I can generate some plots here. So the plot right now is showing my R1 substituents versus my R2 substituents. Within the individual cells here, I have an average value of a PIC 50 for target 1. If I'd like to change that, I could see target 2, I could see target 4. If I'd like this to show up a little bit more for the projector here, I could make those colors even more bright. In addition to this, I've got more control over how I'd like to display things. Right? So if I want to do some advanced options, I could change this rather than a grid, I could change it into pie charts, and even change the circle size to say, hey, let's say the number of molecules within there. Change the circle size. So here I can see at position 1, a phenyl, and position 2, a chloro. I've made lots of compounds with that combination of substructures here. And the PIC 50 for a particular target here is displayed there. So in addition to showing this kind of multi-level view of the SAR, I can also trellis. So for instance, if I want to see compounds that have a high activity for one particular target, but perhaps low activity for another one, I can create multiple copies of this and then trellis based on, let's say, my PIC 52 bin. And let's just make this a 1x2. So here what we're seeing is for very low activity for target 2, here is my profile for target 4. So these are active compounds for 4, but inactive. So these are the most selective compounds here against those 2 targets. And as we scroll down, right, so for my target 2 with high activity, it looks like my target 4 has low activity there. Maybe there's a correlation between that. Right. All of this can be panned and zoomed. You can display molecular components on axes as well as interact with the various bits on there. Okay.
Thanks, Tedman. Okay, so now you've seen the basic functioning of Vortex. You've also seen the cheminformatics intelligence built into it, or at least some of it. Let's move on to an example for those of you who are screeners. This is an HTS identification, and this also shows you more of a structured workflow and the use of a set of templates rather than an individual ad hoc analysis, because in this case we would want to replicate a fairly standard procedure as we analyze different data sets. So the problem statement here, as a screener, I need to analyze large volumes of HTS data to identify my hits for follow-up. While I'm doing that, though, I'd also like to identify likely false negatives and also add those into my follow-up data set. The Vortex solution that you're going to see is a multi-step process. Rather than load a file here, we're going to directly access the assay data management system in Dotmatics to load data. If you have a different assay data management system, we can integrate those as well. So don't worry that you may be using something else. Then we're going to go through a guided workflow that starts on that diagram in the top left and walks around the cycle. We're going to use the guided workflow, predefined templates to visualize the data, and then use some selection gadgets to walk the scientists through the process of winnowing out and identifying the hits. The next step, though, because we also have access to the chemistry, we can do a cheminformatics analysis to try and find likely false negatives. And in this case, it's fairly simple. We're going to take all of those things that were hits. We're going to find all of their chemical nearest neighbors, and then we're going to take some percentage of the closest negatives to a positive and say those are most likely to be the false negatives. So if we include some percentage of those in our follow-up screen, we might actually identify more hits. So with that,
Okay.
Let's see that one in action.
That's right. So here, previously we were loading files, but what I'm going to do now is actually bring some data in from corporate database. So in this assay analysis component here, I can do a number of things. I could do some comparisons between molecular results across different experiments, but here let's just do an original analysis here. Now this is actually looking at our database. I'm going to pull in some single point data from a particular experiment, and I'd like to analyze percent inhibition. So this is pulling data from the database. I've got a number of things I can do. I can show the individual plates with the heat maps. I can calculate the overall Z prime for the whole campaign. I can calculate per plate Z primes, which is what I'm showing here. So each unique plate ID, there's a Z prime value associated with that. And then I can get some information here as well. So in this column here, my numeric data, this is my response data. If I'd like to calculate the standard deviation there, the median, I've got that information for me. Now, if I'd like to set a cutoff of which results are relevant to me, I could add a standard deviation cutoff and start whittling this down interactively to see, hey, this is my cutoff here. Let's make this a little bit bigger there, as well as exporting here. So all of the data, if you'd like to cherry pick some compounds, all of the well data is associated with this. You could export a file from there. But what we may want to do is take some of the most active compounds here, and then as Rob was saying, we might want to see what compounds in chemical similarity space are nearest to these that we might want to retest, for example. So what I'm going to do now is actually bring the chemical structures in to my table. Right now, the table of data here has my compound IDs, but let's get the structures from the database. Here, let's use the compound ID and select the project we want to do. So this is taking my compound IDs, going to the database, returning with the compounds. So here, let's fix that column. And so here's our molecular data now. And if we see our molecules here that we've selected, we could now do some clustering based on those chemical structures. So let's do that. So let's get clusters around those selected molecules. So what this is going to do is use those selected molecules as the cluster centers and create clusters around those central points. So now we're clustering that data. And the result here will be 4 separate clusters that have distance data associated with it. So distance from the cluster center, so there's the cluster and the distance. And as Rob was saying, we might want to have similar molecules to those hits as well. So I'm going to type here distance, and let's retest the cluster centers as well as getting some similar molecules to those as well. Right. So now that we have those selected, I can select those 11 molecules and bring that back into my corporate system here by posting back to my browser system. It's going to ask me to log in. And now I have my 11 records there. And perhaps now I want to request an assay based on that.
Okay, thanks, Tedman. So that shows you a much more predefined workflow and the kind of visual templates that you might share amongst teams to implement more standard views or more standard procedures. Okay, so for those of you who are in biologics research, who've been patiently suffering through all the small molecule stuff, this one's for you. And this will show you some of the bioinformatics analysis that we have in Vortex. So as a molecular biologist, I want to see and operate on my sequences with their associated experimental data at various scales. But my problem is that most software, particularly visualization software, can't handle the large data volumes that I need, both for display and also for the computation. Vortex solves this problem because, as I said in the introduction, we have spent a lot of time on engineering the back end to ensure that it can handle the very large data volumes. You've already seen that it can combine a scientific object with experimental data. It really doesn't matter if that object's a sequence or a small molecule. And it has the bioinformatics analysis built in to let you understand and interpret your sequences. So Tedman's going to walk through an example now of loading sequence data and using a sequence analysis to compare two data sets.
Right. So one of the capabilities that we have with Vortex is to load extremely large data sets. So here I actually have a separate tab open with a number of sequences. And you can see we're scrolling vertically. We can scroll horizontally as well. And if we take a look in the lower left-hand corner here, the number of sequences that I have open here is 4.7 million sequences. So, right, I don't know how the GoToMeeting is responding, but this is as fast as I can scroll. You can see the data there as well. If I turn off the numbers, I can resize the shape of my table. Again, instantaneous. And this is not just sequence letters, but if you pick on these, it has chemical intelligence. It knows what the underlying residues are. So if you deal with non-natural amino acids, we can depict those and understand those, as well as a number of other annotation capabilities, hotspot identification that we can do there as well. So we can handle very large data sets. I'm going to open a slightly smaller set of this just to work with. Let's open a slightly smaller set just because I'm on a business class laptop. You can see the memory here. I'm going to close that FASTA file. So what we're looking at here is a number of sequences that have been culled from that larger data set, and these are sequences of antibodies that were determined after patients had been given an HBV booster vaccine. So we've got a data set here of 155,000 sequences. I've got additional information here. So the heavy chain CDR3 has been pulled out. And perhaps we want to do some sort of analysis to find out what the most common CDR3 is. So what we're going to do now is we're going to select a subset of this and then do a clustering based on that subsequence there. So let's first look at the length here. So my CDR3 length, here I've got a checkbox picker, and I can take a look and see right? The length of 12 or 13, maybe that's of interest to me. And I'm going to just select those and even create a new window. Maybe I'd like to create a separate workspace for that. So let's create a separate workspace with just those sequences. Now I have 12,000 sequences here and their CDR3s. And what I'm going to do now is do a calculation. So I'm going to cluster all of my sequences based on this CDR sequence. Now I could use a BLOSSOM sequence, but here, since they are the same length, I'm going to do kind of a loose alignment based on how many residues are different. So here I want to identify the most common CDR3, so let's allow one difference to occur within that, and let's link this back to the original one. So we're still 12,000 sequences. So when you do the pairwise correlation there, what we're doing now is actually 1.6 million sequence alignments, and that happened pretty quickly. So now if we take a look at our aggregated table, you can see that we've got a number of clusters here. If you want to see the size of the cluster, we could sort there. And now we've identified right, the most common sequence here. So 467, 10% of the time here, we have this sequence. And you can notice here, right, there are some differences, right? We allowed the system to say, hey, let's keep it in the cluster as long as there's one match or one mismatch or less. Okay. The nice thing about this is we can select the cluster and see back to the original table what that data looks like. Right? We could pass this along and do some further analysis here as well.
Okay, thanks, Tedman. Okay, so the next part of the presentation is to talk more about the integration with the Dotmatics suite. Most of the examples that you've seen so far, we've started by dragging in an SD file or a FASTA file and getting data in via files. A very common use case, though, is I first want to go to my corporate databases, my chemical or biological registry, my assay database, whichever ones. I want to do a search, find some interesting data, and then I'd like to visualize that, make selections. So that's going to be a combination of a product called Browser, which is the search of corporate databases. And again, it doesn't matter if the registry and the assay database are in Dotmatics or if they're in a third-party database system. So the use case that you'll see here is Tedman will do a search, find an interesting set of data, send it to Vortex, use the filtering and selection tools to create a subset, and then send that back to Browser as a new hit list. And then the second example, coming back to chemistry, is to use a tool called Reaction Workflows, which is intended for library design. It's a graphical workflow building tool where you can build up procedures to do things like enumerating libraries by reaction scheme, which is what you'll see today, or by scaffold and R group, to use transformation nodes to do idea generation or to do structure cleanup. So in this case, the example is going to be to build an enumeration of a virtual library. And by its nature, this is an automated tool, but sometimes you want to intervene manually. For example, I enumerate the first step of a library, but before sending that all the products to the next step, I want to intervene and pick a subset, and Vortex is the ideal tool anytime you want to do manual intervention in these. So we're going to start with the browser example, and then we're going to do the reaction workflows example.
Thank you, Rob. So as Rob mentioned, right, the Dotmatics platform here is web-based, so we can access our database through a web-based form here. And I'm going to actually continue on the sequence-based first to show an example. So let's take a look at our project here related to sequences. So maybe we want to take a look at all of our sequences that are in a certain project phase. Right? This is just example data, but here we're going to pull up, in this case, some antibody data from our corporate database. And what we can do here is export data directly from this view and do some further analysis within Vortex. So we've exported data. We've determined, right, there's some sequences there. In this particular example, I've applied a template. So the data isn't coming over as raw data. It's coming over initially as raw data, and then I'm performing a number of visualizations here to help me make my decisions for what to do next. And then we can use, right, the capabilities here to do some filtering. Maybe we like to select some sequences here. I've got 7 selected sequences. The profile looks good, and I can push this back into my web view that I was previously in. So now I have those 7 sequences that I've had refined, and perhaps here I want to submit these sequences to my colleague to run some follow-up assays as well. So this works directly exporting and getting, I guess, visualization templates applied automatically. You can also do a very similar thing with chemical data. So here I'm showing a form, and let's just do a substructure search. Let's see if we can put some query atoms within here, and maybe we want to see all of the molecules that have that substructure and an IC50 less than 10. So let's run a query here. So there's 39 records here that I've returned, some data associated with it. If I'd like to see particular graphs, I could. Additional data gets presented in other tabs in this example here. But similar to what we showed with the sequences, we can directly export these 39 records. And in this case, I had batch information. So I've got 133 batches, right? Because there could be some IC50s against different targets. But it's nice that Vortex can aggregate the tables. So in other words, you could collapse this table down, and we can aggregate based on my ID. And let's just show this aggregate as a first value. So now I've got an aggregated table. Right? So across all of the batches, right, here's my IC50 profile. If I'd like to see the individual 4 IC50s, I could see those. You can change what gets displayed in this aggregated table as well. You could show the aggregate as, let's just say, the minimum value there. So this is the smallest IC50 I've had for this particular molecule. Okay. And again, making selections and then posting back to browser allows you to continue on your workflow, and then maybe request an assay or submit this information to your colleague to take a look at this hit list that you've been working on.
Okay, so now we're going to look at the reaction workflow integration.
So reaction workflows is another element to the software. Let's switch over to that. And for this example, what I'm going to do is import a previously built workflow. And this workflow here is an enumeration for various PI3K inhibitors. So what we're doing here is we've got a node here with a number of reagents. We've got a substructure filter here saying, hey, my secondary amine must be present only once. And then we've got what's considered a vortex node. So this is a node in this workflow that allows us to do some manual interactive selections while this workflow continues on. Down here, you'll see this is our key intermediate that we're going to make a reaction. Here's the reaction, and then we're going to get some results. So if we run this, we've got 3 secondary amines, but only 2 of them have only a single presence of the secondary amine. And as we charge through here, we've got 2 molecules that eventually get virtually created. Now, the reagents here, maybe we'd like to add additional reagents. So let's go to the store here. We've got common reagents from e-molecules. If you have your own inventory, you could search that right here within the application to come up with what available secondary amines do we have on hand. But let's just choose these secondary amines. So now we have 266, and as we run this, right, we have 281 final molecules. But let's go into this node here and take a look at those secondary amines. So here's a list of all of those secondary amines that we loaded up that we're thinking about using in our reaction. Maybe we want to create a subset of this. So let's just do a scaffold tree to determine another way of looking at these compounds. So here I've got the most reduced scaffold saying that there is a single ring within this compound. Here we've got a depiction saying we've got 2 rings that have been connected. Here a fused ring system. So perhaps we want to get a random sample of different graphical structures here. So we could do select a random sample and say from that reduced smiles, let's get at least 5 per group. So now we've made a selection, and if there were at least 5, right, we've got a number of 1, 2, 3, 4, 5 fused rings, 5 single rings, and so on. So if those that subset of reagents we'd like to use, we can now push this back to reaction workflows. And you can see our 28 molecules now have been created. And then as we run this, we now have our 28 products. Now that could be good there. Maybe we'd like to do some more analysis, but if we are going into the lab to actually make these, one of the nice things is that we could now put our electronic lab notebook node into this as well. And now this will actually create an electronic lab notebook experiment for us, load up all of our reagents. We can add some conditions to the reaction and have that ELN experiment ready for us to actually perform the chemistry.
Okay. Thanks, Tedman. So I'm just going to briefly talk about some of the things we're working on for the next release. But before I do that, just a reminder, since we're getting to the end, if you have any questions, please enter them into the questions panel in GoToMeeting, and we'll try and answer those right at the end. So the way we develop Vortex is highly agile. We're on about a 6-monthly release cycle. Everything you've seen today so far is in the the last release, which came out in about March. So the next one will be scheduled for Q3 this year. Most of the projects that we do are driven by one or more customers with a particular need, but then we build everything back into the main code base so that something we develop for a customer becomes available to everybody. Again, this is by no means comprehensive. There's a huge amount of work going on, but I just wanted to touch in a couple of areas that we are working on for the next version of Vortex. So we talked today a lot about small molecule research, and we've also shown you some tools if you're doing antibody discovery. There's an increasing interest in, let's say, molecules in the middle. So a lot of use cases for chemically modified peptides and nucleotides. And coinciding with that interest, there's also, out of the Stoya Alliance, which we're members of, the Helm 2.0 software has recently been released, and with that comes a lot of really nice ideas about how to graphically represent these kind of structures. Because the issue is the two representations you've seen so far, if we look at the diagram on the top left there, if we represent these as the chemical structure, it becomes very hard to see any level of detail about what's going on. If we represent them as a pure sequence, there's a lot of information missing. So the most useful representations are the kind of things you see to the right of that table in these other screenshots, where we have some kind of representation of the oligo and of, in this case, of the cross-linking. So we can really understand the structure, but importantly, all of those three representations are off a single data object. So it's not that we're storing the structure, the sequence, and something else. We have one standard object that we can represent in different ways. So within the next version of the software, we'll have these graphical representations of these kind of structures, and then to go with that, a set of analysis tools, things like common core finding, but also the ability to do sequence alignment on these non-natural sequences, to do regular expression searching, etc. At the other end of the scale on pure sequencing, we're beginning to look at including tools for the analysis of NGS data. So just as you saw with molecules and sequence data, we need a way to get that data in from files, we need a way to display it, and then we need a way to do the necessary calculations and analysis. So to get the data in, we're adding readers for the SAM and BAM files for the alignment maps. One of the issues, of course, we're hitting here is the volumes of data. So this is not the limit. This is just one example that we currently have, where we're loading 1.8 million rows, and then some of those residue widths are up to 250 million, and the software is quite happily handling those. And one of the ways to make sure that this is possible, as it says at the bottom under fast graphics performance, is to do progressive disclosure of the information. In other words, if I'm zoomed all the way out of a 250 million residue width, I really can't understand much detail. So there's no need to actually render much detail in the software. Only as I begin to zoom in, for example, from the top view to the middle view, and you can imagine zooming all the way in eventually to the sequence, only as I hone in on smaller sections of the sequence do I show more information, and that way we can keep the graphics performance consistent, however big the data. So we'll have the ability to read these files and then to display the fragment assemblies and things like coverage maps and variant analysis. So all of that's coming in the next version. If you're an existing customer, we allow you, for development purposes, to access the daily builds that our developers are working on. So not that we'd expect you to put this into production, but you can at least download the work in progress and understand what we're doing and have input on that, and give us feedback as we go along. So with that, thank you very much for your attention. We're almost at the top of the hour. We don't have any questions yet, but if anybody does have any questions, now's the time to type those into the questions window. If you would like more information, or you'd like to share this with your colleagues, this recording will be available in about the next 48 hours. You can also go to our website and look under the Vortex product page. You can email directly, either myself or Tedman, or if you're in one of the cities shown over on the right-hand side, in the next 2 months, we're embarking on a tour, firstly across Europe, later across some cities in the US and Canada, and we'll be introducing all of the Dotmatics software, including Vortex, and introducing some customers to talk about use cases. So with that, I'll hand back to Tina, and we'll see if any questions come in.
Okay, thank
If
you,
not, we'll
Rob.
wrap it up.
Thank you, Tedman. Just to recap, the recording of today's webinar will be on the Dotmatics website in a couple of days. If you have any further questions, you can reach out to Rob and Tedman directly. Thank you all for attending today's webinar, and that concludes today's event.